Hey, you've made it to the end, just a little bit to go. All right, so what happens with these signal transduction pathways? Well, oftentimes signal transduction pathways can do a couple things. Again, either they switch genes on or off to cause synthesis of proteins, or they switch enzymes on or off. So again, this is just sort of a generic signal transduction pathway. The ligand docks on the receptor, that causes the transduction or shape change. You get this transduction pathway where different proteins in the pathway get activated. And in this case, a protein inside the cell gets activated or phosphorylated, and then that causes transcription of a gene, and you start making some new protein. Now, again, sometimes you're making new protein, sometimes you're just activating stuff that's already there. And a cool example of this is seen in what you're going to do in lab. These are shmoos. Now, a shmoo is a cartoon character from like the 50s that had this sort of rounded body and extended head. Super cute. But what can happen is you can get change in shape of yeast, and the change in shape of the yeast is what's referred to as a shmoo. What happens is the portions of the membrane extend out, and you have this sort of extension that resembles a shmoo. Okay, so let's talk about this. Again, this is what you're doing in lab. Yeasts are single-celled organisms, and you can have sort of different parts of the life cycle of yeast. There's an asexual reproduction, and there's sexual reproduction that occurs. Now, with asexual reproduction, you have these one-celled yeast, and they form a little bud, and then it will be two yeasts, and so on and so forth. But when you get two different types or two different strains of yeast, they release those mating factors. We saw this, oh my goodness, all the way back at the start of the lecture. So here are two organisms. One strain is alpha, the other strain is A. They release their mating factors, and then they have receptors that detect the opposite mating factor. Now, what happens when these different mating factors dock on the receptors? This is what we're going to zoom in and look at here. What happens is when the mating factor docks on the receptor, here in this case, you activate a G protein. The G protein activates ultimately through the signaling pathway this formin enzyme. And what that'll do is polymerize actin, which again is crazy. We learned about actin here with muscle contraction, right? The actin proteins, we use it in muscle contraction. We use it in cell division with cytokinesis. You take actin and myosin, and they sort of pinch closed and cause cells divide. So this is just another application of actin. But what happens is this enzyme, when it's phosphorylated, it adds amino acids to, or little subunits of the protein, to actin, and then it adds the protein, and then the protein builds. And as the protein builds, it pushes on the plasma membrane, and then that causes the membrane to extend. So what will happen is protein growth occurs in the direction of the concentration of the increasing concentration of that mating factor. So the proteins basically grow toward the mating factor, and that allows the yeast to grow toward one another. Eventually, their membranes will fuse, and then they become a diploid organism. It's a brand new organism formed by the fusion of these two cells. So what you guys are going to be able to do in lab is use these mating factors to cause the cells to actually rearrange their cytoskeletons and grow toward one another. So all through signal transduction. All right, so signal transduction pathways work in a variety of different ways, right? But there are a couple of big features to look at. One is this idea of signal amplification, right? Within each step, you can activate multiple enzymes or multiple other molecules, and there's a real specificity to this. How do you get this specificity? Well, you can imagine different cells have different receptors. So if you don't have the receptor for a molecule, you're not going to respond to it, right? Additionally, think about what's inside of cells. Different types of cells could have different collections of proteins inside of them. And if you have different collections of proteins, that's going to lead to different signal transduction pathways and different responses. Additionally, and this is something to think about in the what if scenario stuff, if you remove a ligand, you can stop a signal. If you can block the receptor, you'll stop the signal. If you have a mimicking molecule that's similar to the ligand, you can stimulate a pathway. So just think about the basics and then all the what ifs they can apply. All right. So here are some interesting images to consider when thinking about signal transduction pathways. Here we have the same ligand, right? You can have the same receptor, but one thing that can happen is if you have different combinations of molecules inside of cells, that can lead to different responses. So here you can have the same ligand, same receptor in one cell, but because of the molecules present in the cell, you get response one. But different molecules present in a different cell, you can actually get other responses. Here's another interesting interaction of signaling pathways. It's called crosstalk. So here is some ligand receptor transduction, and the cell does some sort of response. But if there could be a different receptor responding to a different ligand, activating a molecule, and then both pathways interact with a molecule from a particular pathway. Now you can imagine this green ligand here, the green circular ligand, if it docks on the receptor and activates this molecule, that could interact with the molecule that's used in the original pathway and alter its expression. If you've thought about or heard about like drug interactions that occur, sometimes if a person is taking one medication, that's stimulating some pathway, right? But if a person takes another medication, that other medication can cause this crosstalk that can either shut down the first pathway or enhance it. So that's why you always need to talk to a pharmacist about what medications a person's taking, because a particular medication is going to stimulate a certain pathway. But combinations of different medicines can either amplify the original pathway or block it. So thinking about drug interactions is something that's really critical in terms of signal transduction. And of course, what if you have different receptors? If you have the same ligand but a different receptor, that could again stimulate different signaling pathways and different responses. Thinking about an application of this, consider adrenaline. What does adrenaline do to the body? It increases your heart rate, it increases your blood pressure, makes your pupils dilate, it causes the breakdown of carbohydrates to release sugar into your blood. It will heighten alertness in your nervous system. It reroutes blood from your digestive system out to your limbs or appendages to get you ready to run or fight or do whatever you're going to do. So how does one ligand cause all those different responses, right? How does adrenaline cause all those different changes in the body? Well, if you have that one ligand, adrenaline, dock on different receptors, so different cells have different receptors, that could lead to different responses. Or again, if we have the same ligand, adrenaline, but cells have different combinations of proteins inside of them, that can lead to different responses. So understanding signal transduction pathways provide a lot of power in modulating or altering responses of our body. Because if you can block signals or enhance signals, you can change how cells respond, and you can change how people feel or behave. Okay. Now, our last little bit here is on apoptosis. Now, apoptosis is sort of fascinating because we think about cells as being necessary for life, but sometimes you have to get rid of some cells. So apoptosis is sort of an organized cell death or cell destruction. With apoptosis, what will happen is the nucleus and the DNA inside the nucleus will get chopped up and destroyed. Proteins inside the cell will get chopped up and destroyed. And then all the destroyed material is packaged in these little vesicles or membranous sacs called blebs. So we're learning about schmooze and blebs, but they're real things. But anyway, what happens is cells will actually cause themselves to be destroyed. Now, that actually does serve multiple purposes. What if you have a cell that has like a leaking lysosome and it could potentially cause some damage in the cell? Well, you got to get rid of it. What if you have a cell that has highly damaged DNA that could cause out of control cell growth? Okay, well, you destroy it. What happens if you have misfolded proteins that could glom together and destroy materials inside the cytosol or cytoplasm of the cell? Okay, you got to destroy it. So there are circumstances in which you have to get rid of cells. So apoptosis is a way for a cell to be like, I've got problems, I'm done. Now, it also is important in embryological development Here, you can see the development of digits in a hand. As the embryological tissue progresses, the tissue between the digits dies off. So because of apoptosis, the tissue between our fingers has disappeared, and now we have these independently moving fingers. And of course, you can see sort of the remnants of that. If you spread your fingers, you see a tiny little bit of webbing in between the fingers. That's where the apoptosis stopped. But again, it provides sort of freedom of movement of digits. So that's handy. Oh, sorry. Didn't mean for that pun to occur. Anyway, what is an example or an application of apoptosis? Now, we use model organisms in a lot of research because they're cheap and easy and they reproduce quickly. So C. elegans is a type of worm, and so they're studied pretty extensively for all sorts of different sort of biological phenomena. But one thing they've worked out in C. elegans is it's one of its signaling pathways for apoptosis. So if we look closely here, these SED3 and SED4 proteins, these are cellular assassins. These are enzymes that will cut up the DNA and cut up proteins inside of a cell and destroy them. So if we look in the second image here, when you activate SED3 and SED4, they act as proteases and nucleases. Again, they destroy proteins and they destroy DNA. Now, why are these normally switched off? Well, it's because they're under the direction of their boss, SED9. So SED9 is sort of like the handler, and these are the Jason Bourne CIA assassins, right? So SED9 is usually active, and it signals to SED3 and SED4, chill out, don't do anything. But if a certain signal transduction pathway is activated here, the release of a death molecule, the death signal is detected. When the death signal is detected, the signal transduction pathway will actually inactivate SED9. If SED9 isn't working, then SED3 and SED4 become activated, and proteins and DNA get chopped up, and then you form these blebs, and the cell is destroyed. So again, you can imagine stuff where you block death signals or you activate this pathway which quiesces or quiets SED9, all sorts of things they could ask with that. So what would cause apoptosis? Again, if you get that death signal, that ligand docking on the receptor. Also, again, cells will potentially recognize if they have damage, significant damage to their DNA, so they don't destroy or cause things like cancer. Or misfolded proteins. Normally, proteins, if they get misfolded, they get recycled. There are little enzymes in the cell or little organelles in the cell where the proteins get digested. But if you get a lot of misfolding, extensive misfolding, that can be dangerous to a cell for reasons we'll talk about later in the year. So those cells will get destroyed. All right, we'll finish with something that is just a little bit outside the curriculum, but I think is really amazing in terms of cellular communication, and it's soils. There are mycorrhizae or fungi that live in soils, and a fungus is basically either a saprophyte or a parasite. Fungi need to get their nutrients from some other organism. So they have these extensions called hyphae. They're basically like protein tubes or protein straws. And what they can do is insert their mycorrhizal hyphae. They can insert their little proteins into tissues of organisms. Again, if it's dead, they can consume the nutrients in the dead organism. Or if it's alive, they can basically hook up to the plumbing of the living organism and extract their nutrients. Now, because these are like little protein tubes or straws, what can happen is the fungi in the soil, the mycorrhizae in the soil, can actually link up to different organisms. And what has evolved is the capacity for plants in community to communicate with one another. So if you think about trees in a woods or trees in a forest, they are connected to one another by these fungi in the soil, these basically protein tubes in the soil. And what trees in a community have evolved the capacity to do is actually communicate with one another by chemical signals. Trees can do things like send a warning signal to other trees. So if a certain parasite enters the forest, they can send signals to other trees and alert them And then other trees have an opportunity to build defense molecules to help fight the pathogen. Trees can communicate even between different species. And one thing that's really amazing that they can actually do is if one tree is sick, other trees in the community can actually send molecules to them. They can share water, they can share nutrients, or they can share proteins that are used to help fight pathogens. So there's unbelievable communication that occurs in broader communities with things like plants. We don't necessarily think about it, but trees can actually communicate with one another through this chemical signaling pathway that started all the way back with bacteria. So soil health is really important in communities. This is sort of an aside, but when people talk about things like organic farming, they think about, we're not using pesticides that could maybe be harmful to people. But another thing about organic farming is that it keeps microbes in the soil alive. A lot of pesticides and herbicides will destroy bacteria and fungi living in soils. So there's less interaction between the plants that are planted like in a field. Now, with organic farming, what happens is those organisms continue to live and grow in the soil, and then that facilitates greater communication between the plants that people are growing. And what that does, the interaction between those plants, is it actually makes them more resilient. So what researchers have found is that in times of drought, plants that are grown organically are more resilient to that drought because of the microbes like the fungi in the soil that can allow the plants to actually cooperate and work with one another. So when we think about organic farming, it's not just about making myself less exposed to certain things. It's also about facilitating healthy soils so that plants can communicate with one another and produce more food for us. I think chapter 11's sort of amazing because you look at this interaction that occurs between cells. There's something that started all the way back with bacteria that allow them to have coordinated responses. And now even within us, we have trillions of cells that can communicate via chemicals and interact with one another and allow us to do things like experience the world and understand what's going on, right? So for you to perceive your reality, your life, it's because cells are able to communicate with one another. So look through this stuff. As you develop questions, let me know. But otherwise, hope you have a great day. Thanks a lot.